So good afternoon, everyone. I am Maggie Wong, and I'm a final year DPhil student from the Department of Physiology, Anatomy and Genetics. My research interest is to understand how brain development and functions are affected by genetic mutations in brain disorder. And I'm going to show you a new approach that we are developing for disease modeling and developing therapies to understand the different groups of brain disorder. So brain development is very important for the proper functioning of brain. When brain starts to develop, it starts with a sheet of cells, and they gradually fold inwards and eventually fuse together to form a tube-like structure. And these tubes will gradually expand, and some of the region as well, some of the region will constrict depending on how cells are produced and how cells migrate to the destination. When the cells reach the destination, then they will become a specific type of cells, which include nerve cells and also supporting cells. And when the nerve cells, we also call them neurons, when they reach their final destination, they will branch out and have their arms extended and connect with each other, and then they can communicate. And that's how it shapes our brain function. So if these processes, they are under control of very sophisticated series of stages of genes and regulation, and when any of these processes they go wrong, the brain development will be affected. And then when they are affected, it will give rise to a series of different developmental disorders. And those will affect our sensory function, affect our cognitive function, and also our motor movements. So there are many approaches and many ways scientists try to understand how brain disorder are affecting the brain function. The first one includes brain imaging, which will give you the overall structural defects of what is affected in the patients. And also, ideally, then pathologists will get brain tissue from human patients, and they can study what is happening at the end point of the disease. But then if we're looking at a developmental disorder, because the brain tissue, they only capture the end points, so we don't really know what's happening during the process. And also, the developmental disorders, sometimes already happen during the formation of embryos. So it's very difficult to obtain, and also there will be ethical issues. So that's the main hurdle for the research into developmental disorder. So there are ways to go around, but they all have some disadvantages and advantages. One way is to use cell lines that are not nerve cells. So it's good that you can understand different molecules, how they interact. But because they're not in the right type of cells, so you don't always see what is happening really in the patients. And of course, there are animal models. Rabbits, mice, rats are the most commonly used models. It's a great model because you can easily manipulate the genetics composition, and then you can observe the disease phenotypes, you can observe how the behavior is affected. But until at the point that many of the drugs that work in animals, they fail in clinical trials. So because of the potential species differences. So what do? We spend so much time and effort using animal models to understand what's going on and develop drugs, but in the end, it doesn't work in patients. So gradually, the field is moving on to using human stem cells. First, using embryonic stem cells. So those cells are the cells you take from human embryos, and they can self-renew unlimitedly, and then they can become whatever cell types that you want them to become in theory. But because they are also from embryos, so difficult to obtain, also ethical issues. So great advancement in the field is that Japanese group and also American group, they found a way to make induced pluripotent stem cells, iPSCs, from adult cells. So very simple. You just put 4 what we call pluripotency markers, and then they will become fully differentiated cells back into the stem cell state, so they can also become any cell types that you want to, and they can be the unlimited source for getting patient samples. Another advantage is that because they are from patients, then they will have the genetic backgrounds, exactly the same as what we see in the patient. And because I'm interested in genetic disorders, so that would be a great advantage that we can understand what is really happening in the patients. So I told you that iPSC can be derived into neurons in particular because I'm interested in brain disorder. And one way is to make them into monolayer. And the advantage of this is that you can monitor individual neurons, how they look, how they develop, but then you can't really study how they communicate with each other. And also you can't look at the overall structure of the brain. So another advance is that you can grow a mini brain in your lab. So isn't it great? Like, these are all mini brains. We call them organoids. So organoids are the structure that they have, the architecture that's very similar to human brain. They have the organized cell layers. They also have the correct connection and network that is formed between cells. So our group is interested in a particular genetic disorder that affects the cerebellum. So the cerebellum is the brain region that controls motor coordination and learning. And our group is trying to establish a pipeline for disease modeling and developing therapy. So from the start, we work with clinicians and geneticists. We get samples from patients, usually skin cells. So when patients go to clinicians, they do they're diagnosed with the disease, and they will get the genetic test to know which gene went wrong. And then, so we got the sample from them, then we program them into stem cells, and then we differentiate them into neurons in the 2D culture or into mini brain. And then we look at what went wrong in the cellular level and also molecular level. And then, because these can all be done in a very systematic setting, then we can use them to screen drugs and develop treatment to treat patients. And I'm going to show you an example of what I did during my DPhil. So during my DPhil, I developed a novel iPSC derived based disease model of cerebellar ataxia. As I said, first we look at the MRI imaging. We see in the ataxia patients there is structural defect in the cerebellum, and it affects how they move, how they talk, and they also have abnormal eye movements. But the lifespan of this patient are not affected, so they still live to around 80 years old without any problem. And we are very fortunate to obtain the postmortem brain tissue from one of the deceased patients. And from that, we can look at what went wrong. So this is a specific type of neurons in the cerebellum. And we compare with the normal people. And we see that there's deficit in the structure of the neurons. So, and then I used the iPS cells to understand the disease mechanism. And finally, we developed a protocol to make the stem cells into cerebellar neurons. And finally, we're going to use that to understand more of the disease mechanism and also develop the therapies for that. So to conclude, I've shown you the latest technology that we use to understand the disease, and also hope that you use that to develop the therapies. And this will be the way to develop the personalized medicine in the future. So I would like to thank my supervisors and all the collaborators, and thanks everyone for listening.